This protocol provides a highly effective bacterial display system for isolation of novel variants of BirA that allows for specific biotinylation of native proteins.
The main advantage of the technique is that it selects BirA variants using high-affinity strep-avidin selection. This ensures that inactive clones are removed, thus creating a highly efficient selection process.
To begin, in a plasmid editor, design the forward primer to include the last 30 nucleotides of the reverse complement of the peptide coding sequence and add the plasmid binding nucleotide sequence to its 5' end. Design the reverse primer to include the first 30 nucleotides of the reverse complement of the peptide coding sequence and add the plasmid binding nucleotide sequence to its 5' end. Then, to set up a 20 microliter PCR reaction, add the reagents in a thin-walled PCR tube. Transfer the tube to a thermocycler and perform PCR according to the manuscript. Next, run 5 microliters of the PCR reaction on a 1% agarose gel to confirm the amplification of a 6 kilobase PCR product and proceed according to the manuscript. Now, to synthesize the mutant megaprimers with BirA hexahistidine forward and reverse primers in a PCR tube, add 1 nanogram of PBAD BirAE CPX with the prepared target peptide sequence as a template. Set up 35 PCR cycles with an annealing temperature of 60 degrees Celsius according to the manufacturer's instruction. Then